Today, we're going to move into Unit 4, which is dividing the cell. Today, we'll talk about cell cycle and mitosis, and lecture 16 will be meiosis. And we're going to start by just sort of generally, but recognizing that cell division is essential in all organisms for both reproduction and growth. So in the single-celled organism shown here in amoeba, you can see that cell division actually was important for reproduction in that we've now just doubled the population size by this cell division. So we had one, it divided, and now we have two cells. In multicellular organisms, it's not only important for reproduction, but for growth of the organism. So in the mammalian example I have shown here, this is actually a fertilized ovum from a human. And this is, within 24 hours, will undergo cell division, and you'll have two cells as shown here. And then, over the course of the next few months, you will have a series of amazing number of cell divisions, and they're really highly controlled and regulated, so that at the end of 9 months, you end up with a multicellular organism that looks like this. And then, if you go a little bit longer and wait a few more years, right, growth cell division keeps going on, and then you have my multicellular organisms, which is my daughter Riley and my son Connor, who are 21 and 24. So cell division occurs throughout life, and we all started out as that single diploid cell. Okay, so cell division continues throughout the life of multicellular organisms, where it's important for growth, replacement, and repair. This is just a picture from your book that shows a cell here that is just divided. It's divided and made an identical copy to itself. And this is in the adult immune system where cell division throughout the life continues to be important for the functioning of a mature immune system. So cell division is highly regulated. It must occur at the right place and the right time. And that's really easy to imagine when you're thinking about going from a single diploid cell into a whole organism 9 months later. But it's also really important in an adult organism that you have to regulate your cell growth. What happens? What's the general disorder called when you don't regulate cell growth correctly? Cancer, right? And so there are alarming statistics that 1.6 million new cases of cancer in 2012 are predicted in the U.S. alone. 600,000 deaths from cancer are predicted to occur in 2012. There has been some progress in both early detection of cancers and curing of some, but it's still a minuscule amount. We still have lots of people that we cannot cure. The overall decreases in frequency over the last 5 years are associated primarily with these 5 cancer types. In men, prostate, lung, and colorectal cancer, there has been a decrease. And in women, breast and colorectal cancer. So there's still really major research efforts that are needed to both do early diagnosis and treatments, and actually care of the individuals that have these. So, most of you know somebody who probably has cancer. All of us have a 1 in 2 to 3 chance of having cancer in our lifetimes. The person I know most closely that had cancer was Andrea Roca. I already introduced you to her. She had pleural pulmonary blastoma type 3, and this was diagnosed in January of 2009. So her mom was working for me, and she came into the office, and she said, and she works with Biochemistry, and she said, you know, Andrea has a terrible cold, and we just can't get rid of it. I need to take some time off, a few days. And so she did, and she came back, and she said, that just didn't help. We're going to the doctor. They went to the doctor. They got some, decongestants, antihistamines. Didn't help. 2 weeks later, she said, I really have to do something. Went to the hospital, and they said, pneumonia. That's what we think she has. Really terrible chest congestion. And she went through 2 weeks or more of antibiotic treatments for pneumonia, and there was no change. When they took a closer look into her lungs and her chest cavity, they found them filled with uncontrolled dividing cells that were just completely filling up her lungs and her chest cavity. So it was compressing her lungs, they were in and around her lungs. So this is a specific type of cancer that is relatively rare. And so, in fact, what I learned from this experience was there were a whole ton of doctors that were important in the treatment of Andrea, but that I already knew. There were so many other people that were important and so many other professions that impacted the lives of these people and both Andrea and her family. So all of these types of doctors, lots of different kinds, hundreds of nurses, literally, ranging from ER nurses to nurses in the outpatient clinic. But the other specialists that I really didn't know anything about were people like child life specialists that actually were able to come in and help her go through radiation therapy without having, it's traumatic, but reducing the trauma associated with that. Therapy dog trainers, never heard of that. That was one of the most important things for Andrea, was having a therapy dog around where she could actually interact with the dog when she was in terrible pain. So my message here is really that the reason why we give you challenging tests, the reason why we want you to learn this material is because the things you're going to be doing aren't straightforward. It's not, I know it's dying and then I finish my problem. It's that you're going to be presented with cases where you're going to have to use deductive reasoning, your analytical thinking skills, and that's what you need to develop here in university for your whatever profession you end up going into. And so I think you really have, I wouldn't be challenging you if I didn't think you were up to it. And we're going to continue to challenge you with these hard problems. And we want you to work really hard at it and take advantage of the resources that we have to start learning how to solve them. Okay, so today we are going to start with mitosis. In sexually material organisms, there's two major cell types. We have somatic cells and gametes. So gametes, what are they? Sex cells, eggs, and sperm. Somatic cells are every other cell type in your body. So if I say somatic cells, or name a somatic cell, it would be a liver cell, or a kidney cell, or a cell in your eye, or a skin cell. Every other cell in your body is a somatic cell. Only sperm in males and eggs in females are the gametes. In somatic cells, these are all diploid. There are 2 copies of each chromosome. One that's inherited from the mother and one that's inherited from the father. And in humans, there are 46 total. In gametes, these cells are haploid. There's a single copy of each chromosome. You would inherit one from your mother or your father. And so, if you have 23 chromosomes, then in a gamete, you might get chromosome number 1 from your mother, chromosome 2 and 3 from your father, chromosome number 4 from your mother, 5 and 6 from your father. So it's a random assortment, and when you talk about meiosis, you will be going over that in detail. Okay, and so how many total do we have here? 23 total. Okay, since we all start out as a single diploid cell, I showed you that on the previous slide, right? That we had a whole baby and organism that came out of a single diploid cell. There must be 2 types of cell division because we get somatic cells and gametes. And those 2 types of cell division are mitosis and meiosis. Mitosis produces all of the somatic cells. This is where 2 daughter cells are genetically identical to the parent cell. So the parent cell divides and produces 2 cells that are identical to each other. They're diploid. They inherit 2 copies of each chromosome. That's what we're going to be talking about today. Meiosis is the subject of the next lecture, and that's for the specialized form of cell division that is for production of gametes. There are 4 daughter cells that are genetically different than the parent cell. They're haploid, right? The parent cell was diploid. They're haploid. They inherit a single copy of each chromosome. And this is often called a reduction division because you started out diploid, having 2 copies of each chromosome, and you end up haploid. You have only 1 copy of each chromosome. Okay, so before we get to the stages of mitosis, let's look at the cell cycle, which describes the cycle of cell growth and division in all cells. And we have interphase and the mitotic phase. Those are illustrated over here, and you can see from this pie chart that interphase, most cells spend most of their time in interphase. The mitotic phase, when you actually have the division of the genetic material and the cell divides, this happens during a very small period of time in the cell cycle. So most of the cell cycle is taken up by interphase, which can be divided into 3 different phases that you have to know. The first is G1, so shown here. This is when there's synthesis of macromolecules and cytoplasmic organelles, everything you need to do to take a cell that started out like this and needs to make a copy of itself. What does it have to do? It has to grow, right? It has to make more of everything, unless it's going to just divide and then make smaller and smaller cells each time, right? So to make a cell that's identical, you not only need to replicate your DNA, but you need to make more of your smooth ER, your rough ER, your mitochondria, your pumps, your lipids, your plasma membrane. All of those things have to be produced, and those are produced in G1. Okay, S phase is where DNA synthesis occurs. This, as you can see, happens before mitosis. That's an often point of misconception, is that DNA synthesis is somehow occurring during mitotic phase. DNA synthesis is occurring during interphase, during the S phase. Okay, and S is followed by G2, which is another growth phase. There are, the cell has a number of checkpoints that are occurring all throughout this growth cycle, where there is the cell won't proceed further into the next phase unless there's a certain amount of mitochondria, a certain amount of rough ER, a certain amount of extra macromolecules. So you'll be learning about that in cell biology. But here, this is the final phase where they're actually making enough so that they're ready for cell division, which is going to occur in mitosis. And the mitotic phase occurs in 5 subphases, and this is when the genetic material is divided. Remember, it was already replicated in S phase, and then the cell itself is also divided. Okay. In S phase, let's look at this a bit more closely. We have replication of the genetic material, or the DNA. So we talked a little bit about the structure of DNA in the last segment, in lecture 9, where you know it's a long polymer made up of nucleotide monomers, right? And a chromosome is a long DNA polymer made up of a linear array of many genes and associated proteins. A gene is a stretch of DNA in a chromosome that has a specific nucleotide sequence that often codes for an mRNA that would then encode a specific protein, such as an aquaporin channel or a Diane. Okay, DNA in S phase is present as in a decondensed form and is called decondensed chromatin. So it's the DNA and its associated proteins, but it's not compacted, it's in a decondensed form. And that's important because in S phase, it has to be replicated, and if you had a really tight compacting of this material, it would make replication much more difficult. So the decondensed phase, decondensed form of chromatin that you see in S phase actually is really important for replication of the DNA. Okay, so after the DNA is replicated, then the chromosome condenses to form structures called sister chromatids. Many of you are familiar with this type of structure down here, right? Sister chromatids, that's what you most often see chromosomes look like. The only time they look like this is when they're in mitotic division. So this is a metaphase chromosome. Most of the time, the chromosomes in a cell are in the decondensed chromatin format. Okay, so this sister chromatid, what it is what we started out with a single DNA molecule, is a DNA molecule double-stranded or single-stranded? Double-stranded, and then it gets replicated. So we have two double-stranded molecules. One double strand is here, and it's identical to this double strand here because it's just a replicate. Okay, so this sister chromatids, this is identical to that. Okay, now this, if you were to unravel this, what this is just DNA. Here's the DNA over here in the base pairing, and it's wrapped around structures, proteins called histone proteins as well as other proteins that are important for compacting the DNA into a very small format, and this aids in the accurate distribution of DNA to the daughter cells. Okay, just a slightly different rendition of this. Here now we have chromosome before we have replication of DNA. Is this what it would actually look like? No. This is shown in compacted form. It's not compacted, it would be in its decondensed form, but this is normally what's shown in your books. So here is a chromosome before replication of DNA. It has a centromere and two arms, one arm here and one arm here. And the centromeres can be in the middle of the chromosome or they can be off-center. Different chromosomes have different structures. Okay, and the chromosome is a double helix. So each chromosome is made up of a DNA double helix with a centromere and two arms. Okay, then we have DNA replication, which occurs when? In S phase, which is part of interphase, and then we form these structures called sister chromatids. Okay. In the sister chromatids, then we have one here and one here. This is one chromatid. This is the other chromatid. These are sister chromatids. So we have two chromatids, two centromeres, and each is a DNA double helix. So, right, each of these consists of DNA double helix. This one and that one. Okay, and then separation of the sister chromatids occurs at division. Then you form two chromosomes. Here we have one that goes to one cell, one that goes to the other cell. Each of them are still DNA double helixes. And so this cell is identical to that cell. This cell is identical to that cell, and these are identical to each other. Okay? So I tried a more complicated demo in the first class and it didn't work at all. So I'm going to do the uncomplicated demo of DNA Velcro chromatin. So here is what I want to represent non-compacted DNA. And so some people have asked me, well, really in a chromosome, what do you have? So we have two arms, one arm here. What does this represent? Centromere, and one arm here. So this one has a short arm and a long arm. And then, if I go look at a finer scale, those of you up at the back can't see, but those of you down here can, I have a sequence of nucleotides, right? A, T, T, C, blah, blah, blah. And how do I know where there's a gene? This is a chromosome. It's got a bunch of genes. How do I know? How can I find a gene? There's a particular start sequence that signals the beginning of a gene. So here it is, ATG. That's the start sequence. You'll be learning about this when you go into DNA, I think it's the next unit. We're going to talk about this. And so there's the start site. And then I read along here, and how do I know when it ends? Because there are stop codons that tell me when it ends. So let's say this gene is encoding a
for eye color.
Okay, eye color. That's a protein that's coding for eye color. Then we read along, and I see TT, CCC, G, CTT, nothing, nothing, nothing. No gene, no gene, no gene, until I come here. Then I know I got another gene starting because I have an ATG. And then we go along here, and we have a stop codon here. Okay, so this is encoding a totally different, this is a different gene, and it's going to encode a different protein. And so we have coding sequence and we have non-coding sequence in the chromosome. And in fact, the majority of the chromosomal DNA is non-coding sequence. The minority codes for genes. Okay, and then these clips on here are meant to represent proteins that are associated with the DNA, because a chromosome isn't a naked stretch of nucleotide monomers that are hooked together. There are a bunch of proteins that are associated with them. And actually the centromere region is DNA and a specific set of associated proteins. Okay? And then when this undergoes replication, this opens up, and what bonds do we break? Hydrogen bonds. Then there are nucleotides that are going to come in here and replicate this, and you're going to learn exactly how that works in the next unit. But so then I replicate this chromosome. So once I've replicated this chromosome, I'm going to have a double-stranded DNA helix here and one here. And this is going to represent then what structure? Sister chromatids. That's exactly right. And so are they identical? This chromosome came from my mother. So, is all the DNA in these sister chromatids from my mother? Yeah, because this one is identical to that. Okay, so that should help you with the problem that we come to again. Back to anyway. Okay. I tried to show compaction with those clips, and it just got all tangled up and I couldn't do it. But basically, then you replicate that Velcro chromosome, and then you compact it into this format so you see sister chromatids. Okay, that was a single chromosome. Let's talk about diploid organisms. We have 2 copies of each chromosome. One that comes from the mother, so it's called a maternal chromosome, and one that comes from the father, so it's called paternal. So these are shown here are homologous chromosomes. This is, let's say, chromosome number 1 that came from the mother, and this is chromosome number 1 that came from the father. These chromosomes, this chromosome number 1 from the mother and father, encode exactly the same set of genes in exactly the same sequence, and they're exactly the same number of total genes in the chromosomes. There might be a slightly different flavor of a gene. There might be an eye color gene that is blue on one and green on the other. But that protein that's encoding for eye color will be at exactly the same place on the same chromosome. Okay. The mother's chromosome undergoes replication and reforms sister chromatids independently of the chromosome that you inherited from your father. And so this is the paternal sister chromatid, and this is the maternal sister chromatid. So these are homologous chromosomes because they are chromosomes that are encoding the same set of genes. Okay, in a diploid cell with 3 chromosomes, let's see if you can identify now what chromosomes that we have. So put a circle around the a pair of homologous chromosomes in this diagram. Okay, here's one pair. Is that the only place you could have put it? Nope. Here's a pair of homologous chromosomes. Here's a pair of homologous chromosomes, and those are before DNA replication. Okay, put a square around homologous chromosomes after replication. Okay, so those are homologous chromosomes after replication. Now put a circle around sister chromatids. Okay, were there any here? Nope, no, none here, right? We only have sister chromatids after replication. So here's sister chromatids, that's another sister chromatid, sister chromatids, sister chromatids. Okay, and then you should be able to then take this starting cell and be able to draw what you'd see in the cells after division has occurred. We have 3 chromosomes, we know that these are, let's say, chromosome number 1. We know these are the homologs, right, because they're the same size. They have their centromeres at the same place. These are the homologs. And so, oh, and that's the centromere. Okay. So this is what you get afterwards. You get one copy, right? These sister chromatids then separate into single chromosomes, and each cell that's resulting from the division gets one copy that came from the father and one copy that came from the mother of each of the 3 pairs of homologous chromosomes. Okay. So in a diploid human cell with 46 chromosomes, in G1 of a number of single chromosomes total, shout out, 46. Remember that came from the mother? 23 from the father. Number of sister chromatids? 0. Very good. Okay. Metaphase of mitosis. Number of single chromosomes? 46. 0. Right? There are no more single chromosomes. You had a chromosome and they duplicated. Number of sister chromatids? Single chromosome and it duplicates. How many? 46 is correct. You start out with 46 and you duplicate it, so you have 46. How many chromatids? 92, because each one is a chromatid. This nomenclature is really important, so you have to know it and you have to be able to work through these types of problems. Okay, so at the end of mitosis, you end up with the same thing as you had in G1. Okay, now we'll go through the stages of mitotic cell division, and the level of detail you need to know what that is what I say in class. Okay, we have interphase. We have our chromatin, our replicated chromosomes that are still in the decondensed phase, and we have duplication of a centrosome. What is the centrosome's function? It's the MTOC. Organizing microtubules, right? And so it duplicates, and now it's going to reorganize these microtubules because they're very important for cell division, and they form the mitotic spindle. So we get duplication of the centrosomes in G2 of interphase. In prophase is the first time we can see the condensed chromosomes. So our sister chromatids are now apparent. They have the DNA has been replicated, but they haven't condensed until this phase. And we have our mitotic spindle beginning to form. What end of the microtubules are here near the centrosomes? Minus ends here, and a plus ends are here. So we're adding to the microtubules. The spindle is forming basically at the center and pushing these apart. So these centrosomes end up at the poles. And so by prometaphase, we have 2 poles with the centrosomes defining those poles. And we have the mitotic spindle, which is forming, that is formed from microtubules that are organized by the centrosomes. We have 2 types of microtubules, kinetochore microtubules, which are interacting with the kinetochores that are on, there are 2 on the sister chromatids, 1 on either side. And so they're interacting with the kinetochores that are located around the centrosome of the sister chromatids, centrosomes. We have non-kinetochore microtubules, which actually interact with each other at the center of the cell, about the equatorial plate, and they are important in actually controlling the elongation of the cell. Okay. By metaphase, we have kinetochore microtubules that have bound to the kinetochores of the sister chromatids, and they line them up, all of the sister chromatids along the metaphase plate. Anaphase, these the microtubules or the chromosomes are separated, the sister chromatids are separated into chromosomes, and this is, let's say, chromosome number 1 from the mother. The replicated one separates, and we have this chromosome number 1 goes back to this pole, and this one to this pole, and let's say this is chromosome number 1 from the father, and it gets 1 copy of the father's goes up here, and 1 copy of the father's goes down here. Okay, so they're lining up independently on the metaphase plate. Chromosome number 1 from the mother lines up independently of chromosome number 1 from the father. Okay, anaphase, they're pulled back toward the poles, they're moved back toward the poles, and then in telophase and cytokinesis, we have reformation of the nuclear envelope. The chromosomes begin to decondense, and we get a cleavage furrow, and the end of mitosis is when these actually separate into 2 cells. Okay, so this single replicated chromosome during metaphase in mitosis contains okay, remember, this was a starting chromosome, single starting chromosome I got from my mother. If I replicated it, does it have DNA from my father too? Some of you might want to change your answer. Okay. Very good. DNA from only one parent. And that is because we're talking about mitosis. Okay, a couple short animations showing mitosis. This cell is going to divide, and you can see even before it divides, it's really highly active. There's a ton of movement going on. It's not just when it divides, and there it goes. Divided. Okay. And then this one is a slightly closer up view. And it shows the dynamic nature of
The movie of dividing plant nucleus. The movie is greatly speeded up. Even so, you can see that the chromosomes take a long time to start moving in an organized fashion.
So they're being pushed and pulled around by the kinetochore microtubules.
During this time, spindle fibers, which you can't see, are growing and breaking down. Eventually, the spindle captures the chromosomes and moves them to the equatorial plate. This is metaphase. When the centromeres divide, the daughter chromosomes can be pulled to the poles of the spindle during anaphase. This movie stops short of telophase.
Okay, so how does this actually occur? Here we have our cell with our centrosomes on either side of the poles, and there are a bunch of kinetochore microtubules that are attached to this side of, here's a sister chromatid, and there's a kinetochore region right here, and the other side of the sister chromatid also has a kinetochore region that's attached to a bunch of other kinetochore microtubules. Okay, and then this sister chromatid, this is going to be separated, and this chromosome then is going to be transported back here, and this chromosome is going to be transported back here, so they end up at opposite poles. We also have overlapping non-kinetochore microtubules, and they're actually adding subunits at their plus ends, and they are important in elongating the cell during this process of cell division. Okay, these are Mitotic-Jacks that students in the past have made for, I thought that that last slide always looked to me like a jack-o'-lantern. Okay, so how does this model of chromosome movement really occur during anaphase? Here is a microtubule, and this is the metaphase plate, the MTOC, or the centrosome of the cell. The microtubule, which pole of the microtubule would be on this side? The minus end, and the plus end up here, right? So we have plus end up here and the minus end out here. So what motor protein would be involved in movement of these chromosomes? Diamine. So that shows you can't forget what you learned before the midterm, after the midterm. Okay, so I have now a demonstration to show you, so if Becca can come down and help set this up, and while she does that, I'm going to go over these, which are my chromosomes. Okay, so I have, this is what structure? Sister chromatids. How many DNA double helices do we have here? 2. These are the centrosomes, and these structures here are the kinetochores. So they are just complexes of proteins that are associated with the DNA in this region, and these have attachment points for the kinetochore microtubules. Okay, so now what do I have here? Homologs, right? So this is homologous pair. You can tell that they're the same because they're the same length. They have their centrosomes in the same regions. And they actually have the same genes. We have the green gene and the red gene. They're at the same place. But the red gene here is a little bit different, right? It comes in a slightly different flavor. So it has the, the pigment that makes blue eyes versus the pigment that makes brown eyes. Okay, so I have another pair of homologous chromosomes that's shown here. So it's spread out pretty far like this. Yeah. Okay, so these guys, these are homologs too, right? What's different about these than those ones? They're shorter. What does that tell you about the number of genes they encode? These encode less genes. So the longer chromosomes encode more genes because it's really just a reflection of the amount of DNA that's in there. Okay, so now what we're going to do, these guys have lined up on a metaphase plate. And so we have, what are these structures here? Centrosomes, good. Pink centrosomes. And what ends of the microtubules do we have? Minus ends here, plus ends up here. And so now I'm going to arrange my chromosomes up on the metaphase plate. Do they go together or are they separate? They're all separate, right? And so they actually get, okay, these guys a little bit closer. So these are attached to these. And so we have these microtubules. So they're attached to these microtubules. Here we go. Okay. So now I need student volunteers to be dyneins. I think I need 8 dyneins to come down and help me to actually go through anaphase. So, anybody want to do this? Yes, right there. We have Halloween candy to help you promote the motor movement. Okay, right in the middle. Way up the back, way up at the back. And there's 3 of you there. Okay, good. Come down. Okay, so now you have to orient. Where does he want to stand? He's a dynein and he's going to help with anaphase. Okay. Going to have to stand up here, right? Going to have to stand up here. Yeah. Okay, good. Okay, you're going to stand there. Very good. Okay, and we need somebody to stand here. You're the dynein for that sister chromatid. Okay, and you're the dynein for this sister chromatid. Okay, sister chromatid here. And the man in the red shirt, come over here. You're the dynein there. And we need one more dynein. Right there, front row. Okay. Okay, so now these guys they can only walk on what? Microtubules, exactly. Okay, and so now they have their sister chromatids that they're going to pull apart, right? And then they're going to walk them in what direction? Toward the minus end. Okay, very good. Drop them. Okay, anaphase done. Very good. Okay, now that way they were organized. Oh yeah, sorry, you have to go over and sign because they're videotaping. Sorry. So now you'll notice when we talk about meiosis, the big difference is how these chromosomes are lined up along the metaphase plate. They're lined up differently in meiosis compared to mitosis. In mitosis, they're lined up independently. Okay, so cytokinesis divides the cytoplasm And in animal cells, we have a cleavage furrow, and it's controlled by a ring of actin microfilaments. So, which motor protein do you think is involved in this process? Again, you have to remember what you learned before the midterm.
Thank you.
Okay, click it if you haven't. Okay, very good. The correct answer is myosin because myosin interacts with actin. Oops. Okay, here's another one. What's moving in this movie of a Drosophila neuron? Here's the cell body and the processes. And this movie was taken by my undergraduate, Nima Hariri, who is also involved in the BioSci Peer Tutor Program. So some of you might have met him. And I really want to encourage you guys to get involved in undergraduate research at your time here at UCI. And so, I'll play it again. Take a guess at what you think is moving. Here are things like right here. That moved. This is moving. Okay. Click it if you haven't. Take a guess. Okay. In fact, the right answer is mitochondria. But the interesting thing is that in fact, it is your dynein motors are moving the ones that are going in this direction. Actually, it's dynein moving in this direction, kinesin moving in this direction, and we've labeled the microtubules with GFP. So, here is your class of 2012 JAC. I just want to say thank you for being a great class. I'm going to turn it over to Dr. Ann here for the next half of the class, and I will see